Okay. Oh, it says live now.
Boa tarde a todos. Vamos iniciar o nosso quinto webinar. Hello to everyone. We are starting our 50th plant pathology webinar today with the lecture by Michael Goodin. Michael is Bachelor of Science. Bachelor of Science in Biology and Chemistry, Brook University. Master of Science and Plant Pathology, and PhD in Plant Pathology by Pennsylvania State University. Today he is professor and University of Kentucky. With all of you, Michael Goodin.
Oh. Muito obrigado. Boa tarde a todos. Muito obrigado por convite. Parabéns, Nefich. Isso é ótima oportunidade. Esta palestra, and I think that's all my Portuguese is used. So I will switch to English. It's quite an honor to be here. Always an honor to be invited by students, particularly. To give a talk. And when Eduardo invited me, I agreed to the talk and I agreed to the topic. Can plant virology tell us anything about SARS-CoV-2 emergence? So I said, sure. And then a month went by and then I actually had to make the talk. And so it's been a very interesting process for me to try to develop those concepts. And I'll try to convince you that indeed plant virology has much to offer in teaching us about virus emergence in general, and we'll look at those parallels. So we will explore how plant virology can inform us about the emergence of zoonotic viruses. Those come from animal reservoirs such as SARS-CoV-2. So this is not so much about SARS-CoV-2 as the plant virology being informed. Some of the concepts for this talk I've taken from my website, the Green Orange Cafe, which I invite you to explore. And thanks to all my colleagues at the University of Kentucky, and thanks to Nefich for all the ideas that went into this talk. So actually, let me just check if I can get a thumbs up. Can you see me okay? You can't read the audience on an online talk. So most people, when they think of an outbreak, it's in this context. You take all the remarkably talented actors that Hollywood has to offer, and you make this absolutely terrible movie, and then everybody thinks that that's how a virus outbreak happens. And I wrote an essay about it that sort of addresses the Hollywood version of virology, if you're interested in reading that. But I think from science fiction movies in general, there's a very important lesson that we can take from this. And that's, you really have to listen to the scientists at the beginning of the movie. The movie will be much shorter and fewer bad things will happen if you listen to that voice of reason at the beginning of the movie. So that's sort of lesson one from the Hollywood version of virology. And another very important thing and sort of relevant, absolutely relevant to our present condition is that when the institutions that are tasked to detect and control pandemic viral diseases are operating effectively, you actually don't see that they're doing any work. So it appears that they have nothing to do because of how effectively they work. Because there's never an outbreak or one that doesn't make the news in great detail. And so outbreaks that do occur fall into distant memory and are perceived as to be no longer a threat, whereas that's actually not true. And so the perception is is quite different when you actually do get an outbreak. But I think a very important lesson to consider is that because we have these surveillance systems and this infrastructure to address emerging viruses, there are a lot of viruses that most of the public have never heard of. Nipah and Hendra, and Hanta and Hunin, Rift Valley. Yellow fever is a distant memory. Argentine hemorrhagic fever. Smallpox is a distant memory. Lassa, even Zika is falling off into memory, as is West Nile. And so emergence is sort of a distant phenomenon to most people until it's not. And this remarkable just emergence and explosive spread across the globe. In January, we had 42 cases reported of SARS-CoV-2, and now, as of end of June, over 10 million cases, and in all countries of the world. And so, there's a couple important things to consider in this, is that we've been here before in human history, and in the cases that have come before, we have two proven solutions to global pandemics of emerged viruses and serious endemic viruses. One is vaccines. So smallpox and polio, measles, hepatitis A and B, yellow fever, mumps, chickenpox and shingles, human papillomavirus, and rabies, largely, and others, largely addressed by vaccines. And then other viruses, not so amenable to vaccines, then have really been targeted by very effective antiviral drugs in the case of HIV or hepatitis C. And that type of approach and control of pandemics I've addressed in this essay, Wisdom That Passes All Understanding. And it's really important for people to understand what came before and what is necessary in present outbreaks to try to control it, right? We have to learn from history. And I think this is where plant virology can start to be a teacher, because what's really interesting in putting this talk together and talking to public audiences over the years, a lot of people don't even know that plants indeed get viruses, okay? And so I have an essay to talk about that, and emergence of viruses in plants and in crops in particular. And my personal connectivity to that is my Uncle Warren in Jamaica, who was devastated by an outbreak of papaya ring spot virus in the late 70s. So absolutely the same phenomena of outbreaks and spread of viruses occurs in plants and cropping systems, just as they do in zoonotic viruses in human populations. So that's a key lesson. The other key lesson is that in response to papaya ring spot virus control, we are 28 years later in, and the transgenic resistance deployed remains effective where it has been deployed and has not been widely utilized, but where it has been deployed, and it's still robust and safe and some of the most stable, cost-effective, and sustainable resistance to plant viruses ever deployed. And we have to keep this in mind when new viruses emerge, and I will talk about that in a second. So we've got to have access to the technologies that are effective in controlling virus outbreaks. This is really critical for message for plant virologists to get across. But one of the things is that since people don't think about viruses in terms of plants, and part of that is a lot of people don't think that maybe they think plants and humans are quite very different, okay? And so if we thought more that we were similar than different, maybe we wouldn't have this separation. And so just how different are we in these plants and humans? So let's play a little game, okay? And we'll try to see where we start to differ or become similar, at what point. So we can start here. As soon as this little computer is thinking whether we should go to the next slide. It's good. Okay. So easy question. Which is the human and which is the plant? Okay, so that's simple. Should be an easy answer. Which is the virus-infected human and which is the virus-infected plant? Okay. Which is the human virus vector or reservoir and which is the plant virus vector? Which is the virus-infected human cell and which is the virus-infected plant cell? And it's probably at this stage that you are not able to distinguish which is animal and which is plant. And that's a really important lesson in that once the virus gets into the cell, then the phenomena of what membranes and how the cell is manipulated start to become very similar indeed. So this is a human cell infected with influenza A virus in the nucleus, and this is a plant cell infected with Sonkus-Yellonet virus, and these are genetically similar in the sense that they are negative strand RNA viruses. So we start to see these commonalities between plants and animals. And so once we get into the cell, it's very difficult to distinguish plant from animal. And so the phenomena of autophagy or manipulation of membranes, or the fact that host factors necessary for replication and recombination are conserved from plants to humans, okay? And the phenomena of genetic bottlenecks and their implications for evolution are similar in terms of the plant examples and the animal examples. And so if I go back to that Smithsonian exhibit about outbreaks, there's a very important statement that gets made. And it says, of course, this presentation was put out before COVID-19 emerged. And the statement is that by understanding how humans have caused epidemics in the past, we can change our behavior now and reduce the risk of epidemics in the future. And I think this is where plant virology has to play a bigger role going forward in the education about outbreaks, because there's this important line. So change our behavior now. And what we've traditionally done in the past is to consider human viruses within just human populations and plant viruses as something separate, and we've done less to show the parallels and similarities between. But if we consider points that drive outbreaks, global transport and travel, living close and high density, changing the land or natural habitats and food production, this applies both to zoonotic virus emergence and plant virus emergence. So let's kind of use, for the remainder of the talk, plant virology effectively as a model system for considering zoonotic emergence. And it's sort of just a mental exercise, and we'll consider if there are legitimate reasons to do that. So the whole beauty of model systems is that they provide you a simpler, more accessible, tractable system that then informs a larger, more complex system. So for example, Mendel's peas, the genetics, the quantitative genetics that were invented in peas applies to all of these other species, humans included, right? Yeast and fruit flies and mice and Arabidopsis are important model systems. And so the key point of model systems is that when you do your experiments in the model system, what's true in the model, okay, then informs the more complex system. And so you can establish the paradigms. And so that leads us to formulating and testing this hypothesis. So the hypothesis then is, if plant virology can inform human virology, then the two should have in common outbreak phenomena, this sort of sudden appearance of viruses in time, a spillover phenomenon, this transfer from wild areas to domesticated areas, common genetics, similar types of viruses should be in play, reliance on vectors or reservoir species for sustaining populations of viruses when they're not breaking out. Okay, so we'll see if those exist in both plant and human systems. And so if this holds, what we then will find is that factors that drive zoonotic emergence also drive plant virus emergence, right? And so factors of mixed infections, genetic variability, adaptation to host drive virus evolution in plant or human systems, and habitat conversion, spatial connectivity, vector dispersal, reservoirs, cultural practices drive ecology, and these two populations then drive emergence. Okay, so we'll see. So let's start with going back to our Hollywood movie and a real case scenario, Ebola virus. When you think of emergence, many people will think of Ebola virus. So Ebola virus first appeared in 1976. And in the subsequent outbreaks, what has been absolutely essential in sort of understanding and containing this virus is to understand what the reservoir species are, how the virus gets into human consumed material, bushmeat of various kinds. Is the virus sexually transmitted, or how is it transmitted? By touching infected bodies. And what is the etiologic agent itself? So that the whole disease etiology, pathology, and epidemiology become essential. And integration of all of this information becomes absolutely essential in containing Ebola. And so in the 24 outbreaks of Ebola, what you find is that we need to know the range of bats, and that these outbreaks occur periodically, and different types of Ebola viruses, because not just one anymore. And the take-home lesson from Ebola virus control, in one of the most recent outbreaks, you have to have rapid deployment, testing and surveys, keep patients hydrated. We've got to isolate infected people in camps and field hospitals. Quarantine becomes very important. And diagnostic tests. Diagnostics, I'll drive this point home often, becomes absolutely essential to knowing where the infected populations are and how to contain them. Okay. And there's a huge parallel to that in plants, where diagnostic labs for virus indexing and diagnosis are essential. And I'll come back to that concept in a second. But so then what happens in this recent Ebola outbreak is that you have emergence, a rapid increase in the number of cases, so-called flattening of the curve, and then a diminished number of cases until the outbreak is reported as being contained and ended. And whether it happens in multiple countries, viruses do not obey borders. You have to have an integration of this containment mechanism across borders, right? And so accurate reporting and international cooperation in all cases. So are there parallels in plant viruses? So let's look at tasboviruses and Geminivirus diseases. Okay, so tomato spotted wilt virus, example tasbovirus, was first described or emerged in Australia in 1919 and is now globally pandemic, okay, or distributed. So what's interesting about tasboviruses, and they'll just use tomato spotted wilt as the virus, it gets us to that genetic relatedness between zoonotic viruses of humans, say Ebola, Nipah, arenavirus, Hendra, hantavirus, or rabies. All these negative strand RNA viruses are genetically similar in many ways to the enveloped ambisense negative sense tasbovirus. So we have this genetic relatedness between human virus and plant viruses. The next thing is, what's the vector? So in the case of Ebola, bats. In the case of tasboviruses, thrips. What's critical to understanding and trying to control tasboviruses is knowing their genetic variability. Okay? And they are quite diverse. And we worry about tasbovirus genetic variation for the same reasons that we worry about influenza virus, because these are two viruses with segmented genomes that can undergo this genetic phenomenon of reassortment. They can switch their genetic segments, okay, to produce new strains. So emergence of new strains of influenza virus are very similar to emergence of new strains of tasboviruses. And whether the virus is a negative strand RNA virus or a DNA virus, in the case of Geminiviruses, like here, these bipartite Geminiviruses, which can switch A and B DNAs or recombined, the population structure of these viruses become absolutely critical to understanding emergence and spread. And both of these viruses have vectors, whiteflies or thrips, and so the population structure and range of the vector is absolutely critical to understanding outbreak and predicting outbreak and controlling it itself. And so in this one case of tomato chlorotic spot virus, we have very parallel phenomena that we see with zoonotic emergence of human infecting viruses. So in the 1980s, this virus was found in Brazil and Argentina, and then uninfected planting material spread. And so we have outbreaks in 2014 in the Dominican Republic and Florida. Weeds become a reservoir, okay, for this virus, which can go into multiple different agronomically relevant hosts, right? So this sort of emergence has a lot of parallels to SARS-CoV-2. So another example of cassava diseases, and I'll focus particularly on cassava brown streak virus, which, although it was first detected in 1936, explosive spread and emergence in the early 2000s. And so for the Brazilians, I just have to say, even though mandioca or cassava is food for about 1.5 billion people in the world, so in Lavras, you might see it as mandioca frita here, which is quite delicious. But on a sort of more global scale, these two major diseases of cassava mosaic and brown streak are highly devastating. The cassava mosaic, Geminivirus, and then brown streak. So what's interesting about brown streak relative to SARS-CoV-2 is that both of these viruses translate a polyprotein which are processed. So the genetics of cassava brown streak virus is very similar to coronavirus biology. There are absolute parallels in the genetics of these viruses. And so in sort of trying to control the pandemic explosion of of cassava infecting viruses where cassava is grown. We know the areas where both viruses co-infect, areas where there's only cassava mosaic disease, and then areas which are currently free of these viruses, which need to be protected from emergence of viruses, outbreaks of viruses in these new areas. Okay, so this is where diagnostics become absolutely essential. And in the case of cassava, this Cassava Virus Action Project is using on-farm next generation sequencing, nanopore sequencing, to do viral diagnostics in field. Has been remarkably successful. And that in combination with having the appropriate engineered resistance. So this virus-resistant Cassava for Africa project has RNA interference transgenics, which are highly resistant. And so this combination of diagnostics and resistant germplasm really, it means great things for the future of protecting cassava, where it is a subsistence crop for many people in the world. Okay. Another third and last example of why we need excellent diagnostics and mapping of viral populations gets into this idea of diseases that are manifest because of synergistic interactions between viruses and co-infections. So in the case of maize lethal necrosis, come on. Yeah, okay, so co-infections are worse than single infections. So the disease itself manifests by infection by maize chlorotic mottle virus and one of several different podoviruses. So you have to know the podovirus prevalent in these areas and the population structure thereof in combination with this other virus. And again, MLN shows, just like Ebola, the outbreak phenomena. First appears in 1974, and then episodic outbreaks over time, some of which have been quite devastating. And so knowing where as many viruses are as possible in any moment in time has become absolutely essential, not just for humans, plants as well, in very parallel ways. And so we sort of the 30,000-foot view of why this is important is in Bahia, Brazil, where you have wild or semi-wild areas here and domesticated areas here converted to farmland. And so the opportunity for spillover, wild to cultivated transmission, if this is not agriculture, it could be human habitation, and it could be Nipah or Hendra virus or something like this, right? And so the phenomena of emergence or spill back from the cultivated areas back into the wild becomes very, very important. So preventing the next epidemic requires knowing what's out there at all times in increasing sophistication. And that requires phenomenal diagnostic labs to do sample collection and surveillance and virus culturing, developing the diagnostic tools, detection kits, basic research characterizing these viruses, sequencing them on unbiased platform, and generating massive databases that are internationally accessible by research groups. And the idea is for every virus, you need to know each variant for every virus in a population across all populations, okay? That's quite a daunting task. And the idea, the sort of take home from that is that you know all the wild plants and you know all the viruses in them, or you know all the human and animal infecting viruses in all species that they infect, okay? And then essentially to to generate this complete tree of virology where you know all virus species in all reservoirs. And sort of as you approach that, that level of sophistication in understanding virus ecology, then you can start to understand areas where there are single infections with sort of local adapted vector populations, and then the predictive risk of spillover or emergence is low, versus mixed infection areas adjacent to susceptible crops where the risk of spillover and emergence is going to be high. So because you need to know this, because there's, for all kinds of reasons we can discuss, there's always going to be another outbreak. And so if we sort of condition ourselves to this reality in terms of plant viruses, then, and we know what to do to contain these and address them, then we're sort of better prepared, at least psychologically as a population, to think about emergence of zoonotic viruses as well. Okay. And so this is, again, coming back to this, that virus indexing and diagnostic labs are just absolutely frontline essential for protecting emergence. And yeah, I've been happy to be a part of several projects like this, both in coffee and in soybean yellow shoot with Antonio Figuera at UFLA. That's been very, very productive. But there's a lot more. And so there's a lot more areas that need to be mapped, and factors that, consider that factors that drive zoonotic emergence also drive plant virus emergence. And so if we revisit the hypothesis, just at the end here, if plant virology can inform human virology, then the two should have outbreak phenomena, spillover phenomena, common genetics in the type of viruses, reliance on vectors and reservoirs. And I think I've given you examples where all of this is true. So we cannot reject this hypothesis. So it's supported for the moment. And we can continue to think about this. And I think it's quite important for us to think about plant and human and animal virus systems as being more similar than different, so that we can learn and integrate emergence and control strategies and inform one another, which we do to some extent. And so with that, I'll be happy to take any questions. And again, parabéns, Nefit, and thank you for the invitation. A lot of the information and source material for this talk was taken from Annual Review of Virology papers, reviews, and it was a remarkable experience to be on that editorial board for 7 years. That is just a really fantastic group and a source of great information when you try to assemble a talk like this together, and you need to integrate across broad spectrums of information. And thank you to all the contributing authors who have written reviews. And with that, I'll be happy to take any questions. Some live comments. Oh, excellent question right out of the gates. Is there any risk of plant virus to human transmission? And so the short answer is no. And so there's no case of human diseases emerge directly from plants. And again, that makes the model system analogy all the better, right? You want a model system that's tractable, that has a low risk of making the researcher sick. So that that's again, that supports that whole sort of model use of plant virology as a model system. Over and above its absolute importance for protecting food supply and agriculture. But, great question. So I got texted a question. Let's see. Yeah. Hello, welcome back.
Hello, Michael. Thank you for your lecture. I have a question about the rapid tests that we see. Each day we see a lot of places that people can do some tests to detect COVID. So there is any test that can be used to detect plant viruses?
Oh, are there tests for plant viruses?
Yeah, for plants.
Yeah, absolutely. So one of the slides I showed for Ebola, if I come back here. If I come back to the Ebola example, there was a little picture of dipsticks. Let's see, this one. So these test strips, and so they're just a little strip of reactive paper, and antibodies are immobilized on them. In effect, they're very similar to like a pregnancy test. So 2 lines positive, 1 line negative, and you can do in-field diagnostics. Then certainly the labs have more sophisticated detection systems based on PCR. For COVID, they're antibody tests as well. Serology is very important. So in the case if you need quantitative data or even qualitative, ELISA for plant viruses is really key and essential. So there are a number of different type of tests, and they're all necessary, but they all have a different type of utility. So under very rapid field conditions, those strips become very easy to use. But the technology advances so quickly. Imagine nowadays we can do screening for cassava viruses in 3 hours underneath a mango tree in Tanzania or Kenya or Uganda or so, using next generation sequencing technology. And that would be unbelievable, even just a few years ago. And so the ability to take that powerful technology under field conditions. But again, that's all driven by the mindset of the diagnostician, in that we need as much information as possible quickly. And the nice thing about the next generation sequencing, that it's unbiased. So it tells you what you think you know, or it might be out there, but it also gives you information in terms of virus discovery in case it's new. And so it tells you what may be new as well, right? So you can capture novel emergence in real time, which is increasingly important.
Okay, thank you. Let's see if we have any questions.
So we have this one about, why do you think it's hard to make a product that can control virus? Oh, like we do with fungus, or okay, so chemical control of viruses. Yeah. Oh, that's a great question. Part of that, in a lot of ways, before we get to the We do that in a sense by not controlling the virus directly, but by controlling its vector. So depending on how the virus is spread, if we can control the vector with insecticides or miticides, in the case of a coffee ring spot virus, then we've effectively, if you control the vector, you control the virus by default. And so in a sense, we do have the same approach. Plants don't have the same type of, they don't have that same immune system. So it's sort of classical vaccine approach, but a potential antivirals. So we're seeing already in the transgenics, that's what the transgenics are, right? Is using RNAi or stimulating that type of response to then use the cell to fight against the virus because it's triggered to recognize viral RNAs. That means deployment of transgenics. And we've been very poor over the last 28 years in deploying this absolutely amazing, effective technology. We are 28 years after the deployment of papaya ring spot virus transgenics, and it's not used where all papaya is grown. And that technology, as effective as it is, has not been employed for other crops. Why are we not protecting tomatoes, for example, or potatoes? We had the technology in 1995 to make potatoes immune to 3 different, the major viruses of potatoes. That technology was never deployed. And so the transgenic resistance to cassava viruses, particularly cassava brown streak right now, is an absolutely important test case, because the genetic resistance, the biology of the virus are one weapon right now, is transgenic RNAi-based resistance. It's the most effective tool we have against these devastating viruses. And I think that will establish acceptance for these technologies and then allow it to be used more broadly in other crops. Because it's getting harder and harder to protect some of the genetic resistance. Tasbovirus is one of the major genes for resistance. SW5 is now broken. And so we've got strains of tasboviruses that are able to overcome the very limited genetic resistance that we have. And so we need these other technologies to kind of keep up with protecting these crops. And it's very similar to what we see with human viruses. In cases where we cannot make vaccines for one reason or another, we've developed antivirals. And HIV and hepatitis C are absolutely great examples of that. And sort of the parallel you see in plant virology is then the deployment of basically a sprayable double-stranded RNA or siRNA that can be sprayed like a fungicide or insecticide that will then target viruses. So we see sort of a convergence of these technologies.
Why do you think it's hard to make products that can control virus like we use with fungus and bacteria, like chemicals?
Yeah, so yeah, that's a really important question. And so we are actually starting. So one of the problems is the deployment. So like I said, we do control the insect vectors with the chemicals. Making an antiviral is a very, very expensive proposition, right? And so we make antivirals for things like HIV and hepatitis C. We will probably not generate antivirals in terms of chemical inhibitors for cucumber mosaic virus, say, or something like that, because of the expense that it would take to develop such. But we have other mechanisms. We have RNAi and other transgenic approaches, CRISPR-based engineering technologies. So it's sort of driven by the genetics of the viruses and the economic potential of how much input you would want to put into making that chemical inhibitor that could be used safely under, millions of acres, and still have it be cost effective. So most of viruses, they're either RNA or DNA genome, so it's pretty easy to target that with silencing technology or CRISPR technology, if genetic resistance. So there are cheaper, easier ways, let's say, to fight viruses directly. Case in point, the virus-resistant transgenics.
This was our last question. So we will finish our webinar. So again, I would like to thank you.
Muito obrigado. Presidente Meio.
If you would like to say anything, we can talk. Okay.
Well, I just to reiterate my thanks and what an honor it is to have such a invitation, and I miss you all very much. Gran saudades. I have a pending proposal to come back to Brazil to do more work like this if it's funded, even under the current situation, we may not be able to come. But if we can keep communication like this, we can at least exchange knowledge and information and keep in touch until such time. And I just, stay safe, do what needs to be done to contain COVID-19. Wear masks, listen to the scientists at the beginning of the movie, because it's serious and it's not a joke. And we absolutely need everyone's participation, as uncomfortable, as impatient as we are. This really requires a grand collective. And in order to control this, it's too serious and too dangerous right now and too volatile to make just personal decisions based on just our own reaction to all of this, because it is a pandemic in a global situation, and we can learn from this, and we will be better next when this is over, as we have learned and should have learned from others, and so we can put that infrastructure in place. But please wear your masks and isolate as much as possible, and do all the recommendations as science advises in this situation. And I hope to see you all in UFLA and Vizosa, and Brazil in general.
We hope to see you too.
Yeah. So thanks for this. I miss you all so much, and so this is a great opportunity to get together. I'm sorry we can't all go for contrafilé and mandioca frita right now. But, yeah.
Okay, thank you, Mike.
Thank you. Parabéns.
Obrigada.
Obrigado a você.